of antibiotic
I
resistance?
think most people do.
Yeah, I thought it was just stubbornness. Like the bacteria just gritted its teeth, if it had teeth, and toughed it out. But looking at the research for this module 4 specifically, it is so much more sophisticated than that.
So much more.
It's not stubbornness. It's an information war.
That is the perfect way to frame it. It's the only way, really. If you think about it, bacteria don't become resistant just by trying harder. They become resistant because they are, and this is not an exaggeration, arguably the most efficient traders of information on the planet. They share survival strategies like teenagers share memes.
It's that fast.
It's that fast and that pervasive.
And that is exactly what we are going to unpack today. For everyone listening, and especially for our learners who are using this as a study companion, we have a very clear mission for this deep dive.
A very specific mission.
We are going to function as your audio study guide. We are going to walk through the source material, Module 4, microbial genetics, evolution, and resistance, and we are going to do it in strict logical order.
We're going to be methodical, super methodical. We won't jump around.
Yeah.
The idea is to build the concept from the ground up. We'll start with the basic hardware, the DNA structure itself,
The building blocks.
the building blocks, and then move strictly through mutations, then to the way genes actually move between cells, and finally get to the specific physical mechanisms they use to break our medicines.
So if you're prepping for an exam, or if you just really want to understand the mechanics of how a superbug is actually built, you are in exactly the right place. We've got detailed lecture notes, we've got diagrams of genetic mechanisms, and some really cool conceptual comparisons to neuroscience that we really need to get through.
It's a dense topic for sure, but the goal here isn't just to memorize a list of facts. If you can understand the underlying logic, the why behind it all, the facts become much, much easier to remember.
Exactly. It's about the story, not just the vocabulary list.
Yeah.
So let's orient ourselves. We need to start at the absolute bottom. The hardware, if you will.
Part 1.
The source material draws a really sharp distinction between two kinds of hardware, the chromosome and the plasmid. Now, in human biology, we just have chromosomes, but bacteria are different. Let's start with the big one, the chromosome.
Right. The bacterial chromosome it's the main event.
Yeah.
In almost all bacteria, this is a single continuous loop of DNA, a big circle.
Just one. We have, what, 46?
We have 46 linear chromosomes, all bundled up in a nucleus. Bacteria keep it efficient, usually just one huge circle of DNA, all folded up in a region we call the nucleoid.
Okay, so no nucleus, just this clump of DNA.
A very organized clump. And this is crucial for the exam and for your understanding. This chromosome contains the essential survival genes.
Defined essential. What does that mean in this context?
Essential means life or death. It means non-negotiable. Everything the bacterium needs to breathe, to metabolize sugar, to build its cell wall, to replicate itself, the core operating functions are encoded right there on that chromosome.
So if you lose the chromosome...
You die instantly. It's not optional.
Okay, so the source uses a great analogy here. It calls the chromosome the operating system.
Which is perfect. Or the brainstem, if you prefer a more biological comparison.
Oh, I like that.
Think about your own brainstem. It controls your heart rate, your breathing, your basic consciousness. If you lose your brainstem, it's game over. You cannot function.
It's the baseline.
It's the baseline for existence. That's the bacterial chromosome.
But then we have these other little guys, the plasmids. And looking at the diagrams in the source material,
you can do it.
these look like
Let's
tiny
work
little
today
circles
to make
of
tomorrow
DNA
a
just
better
floating
day.
around next to the big chromosome. They look
You can
almost
do it.
insignificant.
Let's work today
look
to
small,
make
but
tomorrow
they
a
are
better
mighty.
day.
Plasmids are small circular
You can
DNA
do it.
molecules that are
Let's
physically
work.
separate from the chromosome. They float independently in the cytoplasm. And here is the key distinction you absolutely have to nail down.
What's that?
Plasmids are not essential for survival.
So a bacterium can just live without them.
Perfectly fine. A bacterium without any plasmids can eat, it can grow, it can divide, and live a happy, normal bacterial life.
Right.
But it might be a boring life, and it might be a very vulnerable life.
Okay, so the analogy in the module here is software updates, or, and I like this one better, DLC, downloadable content.
Which is perfect, isn't it? Think of a plasmid as an optional plugin for a video game. You don't need the Flying Mount expansion pack to play the game, but it sure makes getting around a lot easier.
And it might save your life in a tough spot.
Exactly. Plasmids encode advantages. They're bonus features.
And what kind of advantages are we talking about here? What is the payload that these things carry?
Generally, it's three main things. First, and most famously, antibiotic resistance. Second, toxins, the actual weapons that make bacteria dangerous. And third, weird metabolic tricks.
Metabolic tricks.
Yeah, like the ability to digest a strange food source that other bacteria can't touch, like crude oil or certain plastics.
Wow. So the chromosome is how to be a bacterium, and the plasmid is how to be a superbacterium.
Precisely. That's the takeaway. And because they're separate little pieces of software, they can be swapped, they can be treated. But before we get to the swapping, which is really the scary part, we need to look at the structure of a plasmid itself.
Right.
The source material has a specific diagram, a plasmid vector, that we need to break down because this is the absolute workhorse of modern biotech.
Yeah, I'm looking at that image now. It's a circle with a few key-colored blocks on it. Let's decode this for the listener piece by piece. At the bottom, there's something labeled ORI. ORI.
The origin of replication. This is non-negotiable. It is the most important part of the plasmid.
Why?
Well, think about it. The plasmid is separate from the chromosome. So when the cell gets ready to divide and copies its main chromosome, how does it know to also copy this little extra circle?
Oh, right. It needs its own instructions.
It needs its own license to copy itself. The ORI is a specific DNA sequence that acts as a landing pad for the cell's replication machinery. It's a little flag that says, hey, over here, start copying here.
So if a plasmid, for some reason, doesn't have an ORI,
It's dead in the water. When the bacterium divides, the chromosome gets copied and passed on, but the plasmid gets left behind. Within a few generations, it's lost from the population.
so the ORI ensures the plasmid gets passed to the kids.
It ensures inheritance. And interestingly, the type of ORI often determines the host range. Some ORIs only work in E. coli. Some are broad range and work in lots of different species. It's like a software compatibility key.
That makes sense. No ORI, no copies. Okay, moving around the circle, there's a big red section called the selectable marker.
This is huge for lab work, but it's also the core of its function in nature. In nature, this is usually the resistance gene. But think about it from a scientist's perspective for a second.
Okay.
You're trying to insert this plasmid into a billion bacteria in a test tube. How do you know which ones actually took it up? It's microscopic. You can't just look and see.
Right, they all look identical.
So you put an antibiotic resistance gene on the plasmid, let's say resistance to ampicillin. That gene is your selectable marker. Then, after you try to get the bacteria to take up the plasmid, you dump ampicillin into the tube.
Oh, I see. It's brutal.
It is. It's ruthlessly efficient. You kill everyone who didn't get the update. The only things that grow on your Petri dish afterwards are the ones that are successfully holding the plasmid. You've used the marker to select for the winners.
It's a survival test that you engineer.
However,
Okay, then there are these other two regions right next to each other. The promoter sequence and the MCS, or multiple cloning site.
these are the tools for gene expression. They're what make the plasmid useful for carrying new genes. The promoter is like the on switch.
The on switch.
Yeah, it's the sequence where the machinery binds to start reading a gene and turning it into a protein. Without a promoter, a gene is just silent code.
So it's the start reading here signal.
Exactly. But the multiple cloning site, the MCS, that is the really clever bit of engineering. It's a stretch of DNA that has been designed with a whole bunch of very specific, unique sequences that can be cut by things called restriction enzymes.
It's up by enzymes.
Think of restriction enzymes as molecular scissors. Each one recognizes and cuts only one specific DNA sequence. The MCS is designed to be a cut and paste zone. It's packed with a bunch of different cut sites right next to each other. It's basically a universal adapter that says, insert your gene here.
So if I, as a scientist, want to make a bacterium produce insulin or spider silk or that glow-in-the-dark protein,
You take your insulin gene, you find a restriction site in the MCS that isn't in your gene, you use that specific enzyme, those scissors, to cut open the plasmid at that spot. Then you paste your insulin gene right in there.
and the bacterium just goes along with it, doesn't know the difference.
It has no idea. All it sees is a promoter, the on switch, and a gene next to it. So it just starts cranking out insulin protein like it's its job. That is the absolute basis of modern biotechnology.
Wow.
But in nature, these regions allow bacteria to shuffle and acquire various genes effectively. It's a modular system.
Okay, so that's the hardware. We've got the OS, the chromosome, which keeps you alive, and we've got the DLC, the plasmids, which can give you superpowers. But for evolution to happen, for new traits to even appear in the first place, this hardware has to change.
It has to have variation.
Right. So we need to talk about mutation.
Part 2 of our outline, the raw material of all evolution.
The definition here in the module is simple but very strict. A mutation is a permanent change in the DNA sequence.
Permanent is the key word there. DNA polymerase, the copier enzyme, it makes mistakes all the time, but usually they get fixed. The cell has proofreading systems.
Like spell check.
Exactly like spell check. If the mistake is fixed, it's not a mutation. A mutation is when the typo slips through the cracks, gets etched into the code, and is then passed down to the next generation. That's when it becomes permanent.
The source material lists several major types of mutations based on a diagram. We need to visualize these because they aren't all created equal in terms of their effect. Let's run through them. First up, deletion.
Pretty self-explanatory. A segment of DNA is simply removed. It's like tearing a page out of the instruction manual.
And that's usually bad.
Usually this breaks something important. If you delete a critical gene, the cell dies.
Okay. Next is duplication.
A segment is copied twice. So now you have 2 pages of the exact same instructions side by side. This one is fascinating for evolution because now you have a spare.
A spare. What do you mean?
You have one copy that has to keep doing the original essential job so the organism doesn't die. But the second copy, it's now free to mutate, it's free to experiment, it can change and evolve a new function without killing the cell because you still have the original as a backup.
Oh, that's really cool. So it's like having a backup file you can just mess around with while the main file keeps the program running.
Precisely. This is often how new gene families and new functions arise over millions of years. It starts with the duplication.
Okay, next one on the list is inversion.
With inversion, the segment is cut out, flipped 180 degrees, and then pasted back in. Imagine reading a sentence and suddenly a phrase is written completely backwards. The cat sat on the mat becomes the mat on tack.
Yeah, that's just gibberish.
It usually destroys the function of that gene completely. The code is unreadable.
Then we have insertion.
A new segment of DNA is wedged into the middle of an existing sequence. This often comes from viruses or from other mobile genetic elements called transposons or jumping genes.
And that would disrupt the gene it lands in.
Absolutely, like dropping a random paragraph into the middle of a sentence.
And finally, translocation.
DNA moves from one location to a completely different one. It's like taking a paragraph from chapter 1 and pasting it into the middle of chapter 10. This can really mess up how genes are regulated, turning them on when they should be off, or vice versa.
The source also specifically calls out frame shifts and point mutations. We need to really distinguish these two.
Okay, this is critical. A point mutation is the smallest possible change. You're changing a single letter. An A becomes a G, for example.
Like a typo in a single word. Cat becomes bat.
Right. It changes the meaning of that one word, but the rest of the sentence structure is intact. Depending on where it happens, a point mutation might do nothing at all. We call that a silent mutation. Or it might change one single amino acid in the final protein. Sometimes that's a big deal, sometimes it's not.
But a frame shift, the notes say this is nastier.
Oh, it's way worse. So you have to remember that DNA is read in groups of 3 letters. We call them codons.
Right, like the fat cat sat. 3-letter words.
Exactly. A frameshift mutation is what happens when you add or delete 1 or 2 letters, not 3. If you delete just 1 letter, say the F from FAT, you don't just lose the F, you shift the entire reading frame.
So it becomes the ATC, ATSAT.
Exactly. Every single 3-letter word after the mutation is now completely wrong. The whole sentence becomes absolute garbage from that point on.
The analogy in the source is great. It's like if you miss buttoning one button on your shirt, and then every single button after that is in the wrong hole, the whole shirt is ruined.
It's a perfect analogy. A frame shift usually results in a completely non-functional protein. It's a catastrophic error.
Okay, so that's the one. But here's the big question. Why are bacteria so good at this?
Yeah.
Why do they seem to evolve so much faster than we do?
The outline calls this the speed factor. It really boils down to 2 things: math and a bit of sloppiness.
Math and sloppiness. I like it.
First, the math part. It's about generation time. Humans take, 20 to 30 years to reproduce? An E. coli bacterium can reproduce in 20 minutes under ideal conditions.
20 minutes. So
Yeah.
in just 1 hour.
In 1 hour, you have 3 new generations. In a single 24-hour day, you can get 72 generations.
72 generations. In human terms, that would take us back to, what, the time of Julius Caesar?
Roughly, yeah. Bacteria do that in a day. That's the first part of the speed.
And the second part.
Population size. You can have billions and billions of bacteria in a single test tube or in one infection. Think of mutation as a lottery.
Right.
If you buy one lottery ticket, you probably won't win. But if you buy 1 billion tickets every 20 minutes, somebody is going to hit the jackpot.
And the jackpot is a useful mutation like antibiotic resistance.
Precisely. It's just a numbers game. With that many cells dividing that quickly, every possible mutation is going to happen somewhere, just by random chance.
Okay, that's the math. What about the sloppiness?
We call it the lack of a proofreading luxury. Human DNA replication is incredibly careful. We have robust systems to check and double-check because mistakes in our cells can lead to cancer or birth defects. We prioritize accuracy.
And bacteria don't.
Bacteria prioritize speed. They copy fast and loose. Their proofreading is, well, it's good enough, but it's not as stringent as ours.
But for them, isn't making mistakes bad? Doesn't that kill the cell?
Individually, yes. Most mutations are harmful or deadly to the bacterium that gets them. But for the population as a whole, it's a brilliant strategy.
How so?
Because mistakes are options, they are variations. 1 in a billion mistakes might accidentally create a protein that can pump out penicillin. And when you have billions of bacteria, that one survivor is all you need to restart the entire population.
This brings us to a really crucial concept in the outline. There's a big warning icon next to this in the notes. The difference between randomness and selection.
Oh, this is the biggest trap. Students lose so many points on this on exam.
And the key takeaway is, never say bacteria decide to mutate.
You must not. You cannot say the bacteria saw the antibiotic and mutated in order to survive. That implies foresight. It implies intent.
They don't hold a strategy meeting. They don't see the danger coming and decide to change their DNA.
They do not. They are single cells. They have no foresight, no consciousness. The mutations are random. They happen before the antibiotic is even applied.
So the resistant one was already there.
Yes, it was already there, lurking in the crowd of billions, just by pure chance. A random lottery winner.
So then the antibiotic enters the picture.
And it acts as a filter. It's a selective pressure. It kills everyone else. It selects the winner who was already there. The antibiotic didn't create the resistance. It just revealed it by killing all the competition.
That is such a critical distinction to make. Mutation is random. Selection is not.
You've got it.
Okay, so mutations create new weights from scratch.
Yeah.
That's the slow way, really.
Yeah.
But if bacteria only pass traits down to their kids, it would still be relatively slow. This is where the information war concept really heats up.
This is where things get weird.
Part 3, horizontal gene transfer.
HGT. This is the paradigm shift. This is what makes microbiology so fundamentally different from, say, zoology. Humans and all animals, we use vertical transfer.
Parents to offspring.
Yeah.
Down the line.
Down the family tree. Bacteria do that too, but they also use horizontal gene transfer. They share genes directly with their neighbors.
Even neighbors of different species.
Yes. Even distantly related species.
That is just wild. It's like me walking up to you, shaking your hand, and suddenly I know how to speak fluent French because you transfer the knowledge directly into my brain.
That is the perfect analogy. And because of this, bacteria don't have to wait for a lucky random mutation to evolve a new trait. They can just trade for it, acquire it.
And there are 3 specific mechanisms we need to cover: transformation, transduction, and conjugation.
The 3 big ones.
Let's start with transformation. The notes call this the scavenging method.
Transformation is the uptake of naked DNA directly from the environment.
Naked DNA, that sounds vulnerable.
It is. It's just fragments of DNA floating around. Usually it comes from the dead.
From dead bacteria.
Yeah, when a bacterium dies, its cell wall often bursts open, a process called lysis. It spills its guts into the environment, its chromosome shatters into fragments, and those fragments just float around.
So the DNA is just debris.
Right. But some bacteria are what we call competent. That's the technical term.
You
It
can
means
do
they have
it.
special
Let's
receptors
work
and
today
machinery
to
on
make
their
tomorrow a
surface
better
specifically
day.
designed to grab
Okay.
these floating DNA scraps,
You can do
pull
it.
them inside,
Let's
and
work
stitch
today
them
to
into
make
their own
tomorrow a
genome.
better day.
The outline uses a
Okay.
neuroscience parallel here.
You can do
Like
it.
scavenging memory fragments from a dead neighbor and integrating them into your own mind.
It's grisly, but it's incredibly accurate. Imagine a soldier on a battlefield. He sees a fallen comrade who has a better shield. He just picks up the shield and uses it.
So if a resistant bacterium dies
Uh-huh.
and its resistance gene is floating in the debris,
A nearby sensitive bacterium can pick it up, integrate it into its chromosome, and boom, now it's resistant. It's recycling the dead to upgrade the living.
that's amazing.
Yeah.
Okay, next up is mechanism B transduction, the viral accident. This one involves viruses, right?
Yes, specifically, it involves bacteriophages, or just phages for short. These are viruses that only infect bacteria. They look like little Leanerlanders from an old sci-fi movie.
So normally a phage attacks a bug. What's the cycle?
Normally, it attaches to the surface, injects its own viral DNA, and it hijacks the cell. It basically turns the bacterium into a zombie factory
A factory for what?
for making more viruses.
Mhm.
The bacterium is forced to make hundreds of new virus particles. Then the cell explodes, releasing all the new viruses to go infect other cells. That's the normal rudolytic cycle.
So where does the accident come in?
The accident happens during the packaging phase. The new virus particles are being assembled. You have the protein head, the tail, and the viral DNA gets stuffed inside the head. But sometimes the assembly machinery gets sloppy.
It makes a mistake.
He makes a mistake. Instead of grabbing viral DNA to stuff into the head, it accidentally grabs a piece of the bacterium's DNA that's floating around in the cell.
It's a packaging error, like a factory robot putting a wrench into the product box instead of the actual product.
Exactly. So this faulty phage particle goes out. It looks like a virus on the outside. It binds to a new bacterium, just like a virus. It injects its payload, just like a virus. But the payload isn't viral code.
It's bacterial DNA from the last victim.
Right.
So the new bacterium doesn't die.
No, because there are no viral instructions in there to kill it. Instead, it just received a special delivery of new genes. It might be a gene for resistance or for a new metabolic pathway.
It's a Trojan horse, but the soldiers inside are actually friendly reinforcements.
Or sometimes they're deadly weapons. The source material highlights the clinical relevance here, and this is a big one. Some of the worst toxins known to medicine. Diphtheria toxin, botulism toxin, the toxins that cause scarlet fever. The genes for those toxins are not native to the bacteria that makes them.
Wait, Wes.
They were brought in by phages.
So the bacteria weren't deadly until a virus gave them the weapon.
Exactly. A bacterium like Corynebacterium diphtheria is relatively harmless until it gets infected by a specific phage that happens to be carrying the diphtheria toxin gene. This process, transduction, is a major driver of virulence evolution.
That is terrifyingly cool. Okay, mechanism C, the big one, the one that notes really emphasize. Conjugation.
The direct link. If transformation is scavenging the dead and transduction is a shipping error, conjugation is a professional, deliberate transaction.
The source calls this the most efficient way to transfer plasmids.
By far. It requires direct cell-to-cell contact. It involves a donor cell, which we call a $5 plus cell, for a fertility positive. This donor contains a special plasmid, the F plasmid, that has all the instructions to build something called a sex pilus.
A pilus. I'm looking at the diagram. It looks like a long tube or bridge reaching out from one cell to another.
It acts like a grappling hook. The donor cell extends the pilus out, it latches onto a recipient cell, and vial cell, and then it retracts, reeling the recipient in.
So it pulls them close together.
It pulls them right up against each other. Then their membranes fuse, and a pore opens up between the two cells, a direct channel.
And then the transfer happens.
The donor cell copies its plasmid, and this is key, it keeps the original, and it shoots the copy across the bridge into the recipient.
So now the recipient has the plasmid. What's the result?
The recipient now has the plasmid, but here is the kicker. Because the plasmid contains the genes to build a pilus, the recipient now becomes an $5 plus dollar cell. It becomes a donor.
Wait a minute. I'm trying to wrap my head around this. It's a pyramid scheme.
It is exactly a pyramid scheme. One resistant bacterium converts a neighbor. Now there are two donors. Those two convert two more. Now there are four, then 8, 16, 32.
It spreads exponentially.
This exponential spread is why conjugation is so incredibly dangerous in a clinical setting. One resistant bug can turn an entire population of sensitive bugs resistant in a matter of hours.
Just hours. That explains why hospital outbreaks can move so frighteningly fast. It's viral marketing, almost literally.
Yeah.
So, okay, we've covered the hardware, the mutations that create variety, and the sharing methods. Now we need to look at what they're actually sharing. What are the blueprints for? Part 4. The mechanics of resistance.
Right. We often just say the bacteria is resistant, but I think a lot of people treat that as a black box, like it's just immune. But it's not magic, it's mechanics. It is physical engineering.
Real-world solutions.
Real-world engineering solutions to a chemical problem. And the source outlines 4 specific ways bacteria fight back against our drugs.
Let's walk through them one by one. Mechanism 1, enzyme destruction.
This is the brute force approach. The bacteria simply produce an enzyme, a protein machine, that physically seeks out and destroys the antibiotic molecule before it can do any damage.
And the classic example here is beta-lactamase.
The most famous one, yes. Penicillin and all its cousins, the beta-lactam antibiotics, they rely on a specific chemical ring structure to work. It's called the beta-lactam ring. Beta-lactamase is an enzyme that acts like a pair of molecular scissors. Its only job is to find that ring and cut it, snip.
And once the ring is cut.
The drug is useless. It's just debris. It can't hurt the bacteria anymore. It's like cutting the fuse on a bomb before it can go off.
So they build a machine specifically to smash our weapon. Okay, that's straightforward. Mechanism 2, target modification.
This one is more subtle, more elegant in a way. Most antibiotics work by binding to a very specific target inside the bacteria, like a key fitting into a lock. For example, penicillin binds to a protein called PBP, or penicillin binding protein.
And what does that PBP do?
Its job is to build the bacterial cell wall. So when penicillin binds to it, it blocks the PBP from working, the cell wall can't be built, and the bacterium falls apart and dies.
So the drug is the key, the PBP is the lock. If the key fits, the lock breaks and the bacteria dies.
Right. So in target modification, the bacteria get a mutation in the gene for the lock. They change the shape of the PBP just slightly.
So the key doesn't fit anymore.
The penicillin key no longer fits into the lock. But, and this is the clever part, the PBP's main function, building the cell wall, still works perfectly fine.
They change the locks.
They change the locks. So you can flood the system with penicillin, but it has nowhere to bind. It just floats around harmlessly. This is the mechanism behind MRSA, methicillin-resistant Staph aureus.
So that's what's happening there. Okay, mechanism 3, efflux pumps.
I love this one because of the pure engineering involved. An efflux pump is an active transport channel, a pump that's embedded in the bacterial membrane. Its job is to recognize toxic molecules on the inside of the cell and physically throw them outside.
It's a bouncer at a nightclub.
It is a bouncer. The antibiotic manages to get past the door and get inside, but before it can find its target and cause trouble, the efflux pump grabs it and ejects it. You're out of here.
So does this cost them anything? It sounds like it would take energy.
Yes, and that's a crucial point we'll come back to. This is active transport. It burns ATP, the cell's energy currency. It costs a lot of energy to run these pumps 24/7. Resistance is not free.
Good point. Okay, and the last one, mechanism 4, reduced permeability.
If efflux is throwing the troublemakers out, reduced permeability is locking the front door in the first place. This is especially important for gram-negative bacteria
Why them specifically?
because they have an outer membrane, an extra layer of defense, and that membrane has protein channels called porins. These are the little doorways that nutrients and also drugs use to get inside.
The source mentions the bacterium Klebsiella here.
Yes, some highly resistant strains of Klebsiella have a simple solution. They just stop making certain porins, or they get a mutation that makes the porin channel narrower.
So the drug can't get in.
The antibiotic molecule is now physically too big to fit through the hole. It just bounces off the outer hull of the bacterium. It can never get inside to do its job.
Amazing. So let's recap the 4. They were either smashing the drug with an enzyme, changing the target so the drug can't bind, pumping the drug out as soon as it gets in, or just locking the door so it can't get in at all.
Exactly. And remember that neuro analogy from the source. Resistance is not armor, it's rewiring. They are physically changing their internal architecture to survive.
Okay, so now we know how they do it. But where does this happen most intensely? We need to talk about the battlefield. Part 5, environmental context, specifically the hospital.
The source calls the hospital an evolutionary incubator, which is the absolute last thing you want a hospital to be. But if you think about the conditions, it's the perfect description.
Why is the hospital so perfect for creating superbugs?
It's a perfect storm of 3 factors. First, and most obviously, high antibiotic pressure. We use tons of powerful drugs there. This kills off all the weak, sensitive bacteria, leaving only the super-resistant ninjas behind.
That's strong selection pressure.
The strongest imaginable. Second, vulnerable hosts. You have patients with weakened immune systems, with open wounds, with IV lines and catheters. They are easy targets for colonization.
And the third factor?
Close proximity. You have lots of patients, lots of healthcare staff moving between them, lots of shared surfaces. This creates the perfect opportunity for conjugation, that direct cell-to-cell contact we talked about.
So you have the intense pressure to select for resistance, and you have the perfect environment to share the resistance genes once they appear.
It's the perfect storm. Selection pressure plus gene sharing equals rapid, explosive adaptation.
The source also mentions biofilms as a bunker strategy.
Yeah.
Now, I usually think of biofilm as just slime. the gunk on your teeth in the morning.
It is that, but it's so, so much more. A biofilm is a highly organized multicellular community, bacteria attached to a surface. It could be a catheter, a heart valve, an artificial hip, and they start secreting a protective matrix.
The notes call it a self-produced matrix. What's it made of?
It's a complex goo. It's made of proteins, long sugar chains called exopolysaccharides, and even extracellular DNA. It builds a physical fortress around the community.
And the source also mentions something implied here called quorum sensing. They're talking to each other.
Yes, that's what makes it a community. They don't just pile up randomly, they release chemical signal molecules into the environment. They're basically asking, hey, how many of us are here? Once the signal reaches a certain concentration, a quorum, it's a trigger.
A trigger for what?
They all activate the slime genes simultaneously and start building the bunker together. It's a coordinated effort.
And why is this such a problem for treating infections?
For a few reasons. One, the slime acts as a physical shield. Antibiotics literally can't penetrate deep into the matrix to reach the bacteria at the bottom. But maybe even worse, inside the biofilm, the bacteria are packed together shoulder to shoulder.
It's a hotbed for conjugation.
A hotbed for horizontal gene transfer. They are sitting there, protected from our drugs and our immune system, just swapping resistance plasmids like trading cards.
And what about the dispersal stage mentioned in the diagram?
Eventually, the biofilm matures and chunks of it will break off. These are called persister cells. They float away to start a new infection somewhere else in the body. This is why biofilm infections are so chronic and hard to eradicate.
Do you give an example?
A patient with an infected artificial hip. You treat them with IV antibiotics, they get better. You stop the drugs, and a week later, the infection comes roaring back. Why? Because you never killed the bacteria hiding in the bunker. Often, the only cure is to physically go in with surgery and remove the implant.
That is brutal. The source connects this idea to the bacterial growth phases. We have a graph here: lag, log, stationary, and death.
This is crucial for understanding when evolution happens most efficiently. The log phase, or exponential phase, is when they are growing fastest, doubling every 20 minutes. This is when DNA replication is at max speed.
So that's when the replication errors, the random mutations, are most likely to happen.
Exactly. Log phase is the prime window for new mutations to arise. But then comes the stationary phase. Nutrients start to run out, waste products build up, there's no more space. They stop growing.
The source says stress proteins active here. What's the significance of that?
This is the system's insight. Stress is a catalyst for evolution. When bacteria are starving or stressed out in the stationary phase, they turn on a set of stress response genes. And some of these genes encode for sloppy, error-prone DNA repair mechanisms.
Wait, they start making more mistakes on purpose.
In a way, yes. They get desperate. They essentially start gambling, hoping that one of those new mutations will be a solution to their problem. They also become more competent to pick up naked DNA from the environment. Transformation goes up.
So when things are good, they mutate by accident. But when things get bad, they start actively trying to innovate.
That's a great way to put it. Stress accelerates genetic innovation.
Let's move to part 6, toxins and neuroscience parallels. We touched on toxins earlier with transduction, but the source really emphasizes that many toxins are mobile.
Right. The core concept here is that for a bacterium, self and weapon are often two different things. A bacterium might be a perfectly harmless gut inhabitant, but if it picks up a plasmid or a phage that's encoding a toxin gene, it can become a killer overnight.
The examples listed in the notes are terrifying. E. coli 0157.H7.
Regular E. coli is in your gut right now, probably making vitamin K, helping you out.
Right.
But 0157.H7 is the same species. It just picked up a phage carrying the gene for Shiga toxin.
And that toxin is what makes it so dangerous.
That toxin destroys blood vessels and causes catastrophic kidney failure. It's the same basic organism, just with a new weapon app installed.
Scalded skin syndrome.
You can
from
do
Staph
it.
aureus.
Let's
Caused by an
work today to
exfoliative
make
toxin
tomorrow
that's
a
usually
better
encoded
day.
on a plasmid. It
Okay.
literally dissolves the glue
You
that
can
holds
do
your
it.
skin cells
Let's
together.
work
And
today
flesh-eating
to make
strep.
Same
tomorrow a
story.
better
Toxic
day.
shock syndrome.
Okay.
These are caused by
You
superantigens,
can do
often
it.
brought in by
Let's
mobile
work.
genetic elements. The huge implication here is that virulence is a modular trait. A harmless bacterium can become deadly without changing species.
And the neuroscience crossover. The source makes a big deal about information storage and plasticity. I want to dig into this parallel.
This is a fascinating high-level comparison. Both brains and bacterial colonies are complex, adaptive networks. They process and trade information, but there is a massive fundamental trade-off between the two systems.
And then that trade-off is speed versus stability.
Exactly. The human brain, our neurological network, trades speed for stability. We have neuroplasticity, we can learn new things, but we don't rewire our entire personality or our core memories overnight. That would be chaos.
We need stability to function as a complex organism. If I woke up tomorrow with a totally different brain structure, I wouldn't be me.
Right, we preserve our core identity, but bacteria.
They trade stability for speed.
All day long, they are single cells. They don't have a complex identity to preserve. They are more than willing to scramble their own code, to take up foreign DNA, to risk death, and to fundamentally change their genetic makeup within minutes just to survive the next hour.
And the clinical insight here is that this is why infections can evolve faster than our therapies can keep up.
We are fighting an enemy that is willing to burn its own house down just to find a fire exit. We simply can't invent, test, and approve new drugs as fast as they can download new updates.
This leads us perfectly into the final section, Part 7, Studying for the Exam. We need to solidify this for the learner. The outline provides some causal chains to help with memory retention.
Let's do it.
Let's walk through these as if we are simulating a clinical case or an exam question.
Perfect. These are your if-then statements for the test.
Okay, scenario 1. A doctor in a hospital ward overprescribes broad-spectrum antibiotics to all their patients. What's the chain?
The chain is high antibiotic pressure leads to intense selection. The weak, sensitive bacterial die off. The few that are already resistant survive. This leads to resistance dominance in the population. Remember, the pressure didn't create the mutation, it just selected for it.
Got it.
Yeah.
Scenario 2. A resistant staph bacterium meets a sensitive staph bacterium in a patient's surgical wound.
The chain. Close proximity allows for pilus formation. This leads to plasmid transfer via conjugation. The result: a population-wide resistance emerges very quickly.
Scenario 3. A patient has an indwelling urinary catheter for 3 weeks.
The chain. A foreign surface provides a place for attachment. This leads to biofilm formation. This, in turn, leads to protection from antibiotics and the immune system, and it increases the rate of gene exchange inside the biofilm. The result: a chronic, difficult-to-treat infection.
Last one. Scenario 4. A harmless throat bacteria gets infected by a virus.
The chain. A phage infection leads to the accidental insertion of toxin genes via transduction. The result: a sudden increase in virulence. The harmless bacteria might now cause something like scarlet fever or diphtheria.
Those chains are super helpful. Now let's destroy some common misconceptions. These are the things that will get you a big red X on the exam. Myth number 1. Resistant bacteria are stronger.
False. So false. Resistance often comes with what we call a fitness cost. Think about those Afflux pumps we talked about.
They burn energy.
They burn a lot of energy. If you take away the antibiotic, the resistant bacteria are often weaker and slower growing than their sensitive cousins because they're wasting precious energy running these pumps they don't currently need. They aren't stronger. They're just specialized for a toxic environment.
So if you remove the antibiotics from the environment, the sensitive ones might actually outcompete them and take over again.
Yes, over time. That's the entire principle behind antibiotic stewardship, using fewer antibiotics. It lets the weak but treatable bacteria come back and dominate the population.
Okay. Myth number 2, and this one is controversial. Stopping your course of antibiotics early causes resistance.
This is a tricky one. The conventional wisdom is to always finish your course. The logic is that if you stop early, you might leave behind the partially resistant ones, the ones that needed a full 10-day course to be killed, but you only gave them 3 days. They can then regrow.
But is that what causes the resistance?
Not fundamentally. The use of the antibiotic in the first place is what selects for resistance. It's all about how the usage acts as a filter. Overuse selects for high-level resistance. Underuse might select for low-level resistance. The whole picture is more complex than the simple slogan suggests.
Final myth. Myth number 3. Mutation, adaptation.
No. Mutation is random variation. It is rolling the dice. Adaptation is the result of natural selection acting on that random mutation. You mutate first, you adapt if and only if that mutation happens to be beneficial in your current environment.
So mutation is the raw material, but adaptation is the finished product, shaped by the environment.
Beautifully put.
And the core rule from the source material to rule them all.
Antibiotics kill organisms. Evolution kills strategies.
Say more about that.
You can kill a single resistant bacterium with a different drug? Sure. But if that bacterium already passed its resistance plasmid to a neighbor via conjugation, you've killed the organism. But the genetic strategy, the plasmid, has escaped. It survives.
That is profound. Evolution kills strategies.
It's the only way to look at it if you want to win. We aren't just fighting a bug. We are fighting a process of information exchange that is billions of years old.
So let's wrap this entire deep dive up. We started with the simple circular chromosome, the core operating system. We saw how plasmids act as dangerous, shareable DLC updates.
We did.
We watched them trade these secrets via transformation scavenging from the dead, transduction viral accidents, and conjugation, a direct, deliberate swap.
And we saw what those secrets were, the mechanics.
Right. They build pumps, they smash enzymes, they change the locks, and they hide out in biofilm bunkers.
It really is a journey from the smallest possible level, a single change in a DNA letter, all the way up to the global public health crisis of hospital superbugs.
The source leaves us with a great memory lock phrase to end on. Antibiotics are weapons, genes are strategy, evolution always plays the long game.
It's a powerful reminder that we can't just bomb our way out of this crisis with more and more powerful drugs. Eventually, we have to outsmart the strategy itself.
So here is a final thought to chew on, something that's not explicitly in the notes but is implied by everything we've just discussed.
Okay.
If bacteria can so easily share genes across what we would call different species, if an E. coli can give a resistance plasmid to a Klebsiella, then does the concept of a species even really exist for them in the same way it does for animals?
That is the question that keeps microbiologists and evolutionary biologists awake at night. Maybe there are no distinct species. Maybe the entire bacterial world is just one giant global liquid superorganism that is constantly remixing and reinventing itself.
And on that slightly terrifying note, we will let you process that download.
Good luck with the studying.
Study hard, it'll click. We'll see you in the next deep dive.